脑人炎。
大家早上好和晚上好，我是江敏清，我目前在 MIT 做博后，主要研究方向是关于自闭症的 biomarker 生物标志物以及基因治疗的研究。今天由我来主持杨柳博士在脑人炎第88期的一个讲座。杨柳博士毕业于中科院神经所，导师是张旭老师。然后在毕业之后加入 MIT Pikawa 实验室，Pikawa 研究所做Learning and Memory 方向的研究。他是在 Glory Choi 实验室做的博后，然后现在是 promote 到 Research Scientist。我们今天呢，杨柳博士会给我们带来他最新发表在 Cell 上的研究。在我们开始正式讲座之前呢，我们先说一下，在我们今天的讲座过程当中，我们是会对会议进行视频录制的形式，然后欢迎大家在中间提问，然后在杨柳博士全部讲完之后，我们会进入到提问的环节。在正式提问环节过程当中，我们会把录制的部分后面会剪掉，然后欢迎大家随时进行讨论。那么现在我们欢迎杨柳博士开始正式的讲座的环节。
首先感谢大家能参加我今天这个讲座，然后要感谢脑人炎的各位小伙伴啊，忙前忙后帮我撑了这么一次安排这次讲座，然后要感谢刘鼎把脑人炎这么好的一个平台介绍给我，最后要感谢明清，算是临危受命被我们拉回来充当这个主持人，然后我要出一个练习的目的，所以我今天基本上是会用英语来完成这个讲座。不过如果大家提问的时候，可以用自己方便的语言。OK， 那我现在就开始了。So the title of my seminar today is Balance and Breakdown Regulatory and Pathogenic Neuroimmune Crosstalk. From the title, you may see the topic I'm going to address is about neuroimmunology. Neuroimmunology is a relatively old research field. Back to the 19th century, the physicians had discovered these uneven distribution of cellular aggregates and lesions in the brain from patients of multiple sclerosis. Nowadays, researchers have identified that these lesions are actually infiltration from peripheral immune cells into the brain of MS. And with more advanced imaging and like histology technologies, researchers are now able to identify the subtypes of these immune cells. Another major discovery in the field of neuroimmunology is the discovery of inflammatory reflex. In the condition of inflammation, peripheral immune cells release excessive amount of cytokines, and these cytokines are signals via the afferent branch of the vagus nerve to the brain. And after the brain processes this information, the brain further sends signals via the efferent branch of the vagus nerve to affect the peripheral visceral organs, such as spleen. And in the spleen, neurotransmitters such as noradrenaline and acetylcholine are mobilized. And therefore, these transmitters will inhibit the macrophage from further release more cytokines. In this way, the inflammatory reflex can be contained in a controllable range instead of completely going crazy. Well, apart from the indirect communication mediated by vagus nerve and in extreme conditions such as neurodegenerative disease or brain trauma, the central nervous system has long been thought to be completely segregated from the immune cells in the periphery. So the brain has been considered to lack or have very limited amount of immune responses in there. People even give this phenomenon a name, which is called the immune privileged brain. We can look at this classical experiment conducted in 1920s when the researchers transplanted a piece of tumor into the brain. They see the tumor tissue keeps growing. But when they transplant the tumor tissue together with a piece of spleen graft, which serves as a source of immune cells, the growth of the tumor was substantially inhibited. So this observation suggests, well, indeed, the brain does not have sufficient immune responses to suppress the tumor from growth. So this experiment greatly supports the notion of immune privileged brain. So this kind of immune privilege in the brain or central nervous system are secured by some structural features. One is the presence of blood-brain barrier or BBB. Blood-brain barrier is formed by the functionally specialized endothelial cells, which are connected by this structure, which is called tight junction. So with the presence of tight junction, the circulating immune factors cannot randomly enter the brain parenchyma. And also, there's another structure of the central nervous system which can support the immune privilege there. That is the absence of lymphatic vessels. The brain parenchyma has long been thought to lack a conventional lymphatic vessels. It suggests the brain-derived antigens cannot be efficiently drained into the nearby lymph nodes. Therefore, the adaptive immune cells there are considered blind to the antigens originating from the brain. And this kind of immune privilege in the brain, this kind of arrangement kind of makes sense because we know the neurons have very limited ability to regenerate. So by suppressing excessive immune responses in the brain, the neurons can be protected from excessive damage, because if we lose these neurons from too much immune responses, the organism had a very hard time to retrieve these neurons, so the brain function will be severely damaged. But this is a full story. In this past decade, the field of neuroimmunology has been re-examined, and many old concepts and notions have been redefined. For example, is that true the brain is completely segregated from peripheral nervous system? Maybe not. It is true in the brain parenchyma, immune cells can be hard to be detected. But in these border areas, such as meninges and choroid plexus, people found the presence of diverse subtypes of immune cells there. So meninges is this membranous layers encasing the brain, and choroid plexus is a highly vascularized structure lining the brain ventricles. We can see from these pictures, there are actually various types of immune cells stationing there. So it is conceivable these immune cells may release mediators such as cytokines to affect the nearby neural circuits and thereby influence the brain function. And on the other hand, the meninges also contain this extensive network of lymphatic vasculature. So these lymphatic vessels can collect these brain-derived antigens and transport them to nearby lymph nodes for further immune processing. In this way, the adaptive immune system can learn to capture these brain-derived antigens. Okay, so I have just touched upon cytokines a little bit in the previous slides. Now I need to formally introduce these proteins because these are essentially the main role of my whole presentation. So cytokines are a group of small proteins. They're typically secreted by immune cells to regulate immunity, inflammation, and hematopoiesis. Upon stimulus, some immune cells will release specific cytokines, and these cytokines would act on its cognate receptors expressed by target immune cells. And as a result, a series biological outcomes will be generated. And this kind of communication resembles the communication between pre- and postsynaptic neurons. The difference is neurons use neurotransmitters, neuromodulators, or neuropeptides as a mediator for their communication, not cytokine. But recently, more and more evidences show that actually the neurons also express cytokines and cytokine receptors. And likewise, immune cells also express neurotransmitters and their receptors. So in other words, the neurons can also speak immune language. And likewise, the immune cells can also use neural language for their communication. And this shared molecular toolkits enable the direct crosstalk between neurons and immune cells. So far, I have been trying to convince you that due to the anatomical proximity as well as the shared molecular language, there is this possibility that the nervous system and the immune system can directly communicate with each other. But how does it really happen? So in what circumstances this kind of crosstalk can happen? And how can this influence our bodily function? In our daily life, the most familiar example is sickness behaviors. Sickness behaviors is a series of behavioral changes during sickness. When you catch a flu or common cold, your immune system would be radically activated. And as immune responses unfold, your nervous system will be affected too. That's why you are going to go through a series of behavioral changes. You may feel particularly fatigued. You may lose your appetite. You may be less socially active, or you may have a stronger desire for sleep. And sickness behaviors are highly adaptive and orchestrated behaviors. And interesting, this sickness behavior is evolutionarily conserved. We can see in mammals such as mice and these cold-blooded animals such as frogs, we can all observe similar behavioral changes during sickness. And it should be very easy for us to tell apart which side is a healthy animal, which side is a sick animal. And sickness behaviors are mediated by specific molecules and neural pathways. I will introduce this content in my following slides. Because we do not have enough time to cover all the sickness behaviors, so we chose the one that interested us the most, which is a social withdrawal during sickness. I think it's not uncommon to observe that the sick individuals shows this propensity to leave the animal group and remain by its own. This phenomenon is called self-imposed social withdrawal, which is a kind of a social isolation initiated by infected individuals. This kind of behavior may have its evolutionary benefits because we know social interaction is typically beneficial to animals because social interaction may promote information exchange. The animals may learn the new food sources or the presence of predators through social interaction. However, social interaction also carries risks. It may increase the possibility of pathogenic transmission. So, social withdrawal initiated by sick individuals may not only protect itself because this kind of social withdrawal may provide more space and time for the sick one for rest and recovery, but also it may protect the healthy conspecifics in the same animal cohort. And this kind of social withdrawal during sickness is commonly observed in animal kingdom across different animal species. But it is not well known what molecules mediate this kind of behavior and what is neural circuits underlying this behavior. That's the questions we need to address. Okay, this brings us back to cytokines because cytokines may serve as like messengers transmitting signals between the nervous and immune systems. And indeed, multiple cytokines increases their levels during sickness. That's how we came to this hypothesis that particular cytokines whose levels increase during sickness may enter the brain, access the central nervous system, regulate particular neural circuits, and finally give rise to the behavioral change of social withdrawal. So the first task for us was to identify which cytokines may play a role in this process to induce social withdrawal. We want to examine the direct cytokine effects on neurons. Therefore, we used ICV administration to introduce individual cytokines into the brain of a mouse. And then we tested this mouse's social behavior. For social behavior test, we calculate the percentage of time this mouse receiving cytokine treatment. How much of its time is spent with a conspecific and how much of its time is spent with an inanimate object. We use PBS and LPS as negative and positive controls. LPS, the full name is lipopolysaccharide. This is a component of the cell wall of bacteria. So here it is used as a mimetics of bacterial infection. We can see when negative controls, PBS treated animals spend 70% of its time interacting with a conspecific. LPS treatment significantly reduced the time to only 10%. And at the same time, LPS can suppress the animal's moving velocity. Moving velocity is an indicator of animals' locomotor activity. Then we screened a panel of cytokines, and among them, only this one, IL-1 beta, produced a reduction in both sociability as well as locomotor activity, fully recapitulates the effect of LPS. So, we say maybe IL-1 beta is a cytokine we are looking for. Now that we have a candidate cytokine that may induce social withdrawal, we expanded this cohort size and to verify this result. We characterize the effect of IL-1 beta on social behavior by analyzing the data both minute by minute and cumulatively. We found IL-1 beta induced this progressive decline in both social interaction and locomotor activity. So these results confirmed that indeed IL-1 beta is going to induce social withdrawal, and this kind of social withdrawal is accompanied by a decrease in locomotor activity. Next, we need to ask how the brain can sense the presence of IL-1 beta. Because IL-1 beta binds to its cognate receptor IL-1 and achieves signal transduction and further biological function. So our task is to identify the brain regions that express IL-1, right? Because if they express IL-1, it may have the capability to capture IL-1 beta and transmit its signal. Then we generated this mouse line. We call it IL-1 Cre mouse line. This line expressed a Cre recombinase and a GIP-MYC reporter under the control of IL-1 promoter. So this line can serve as both a reporter line because it's expressed as a GIP-MYC reporter, as well as a Cre recombinase line, which may enable us to achieve genetic manipulation in IL-1 expressing cells. First, we use in situ hybridization to co-label the transcripts of IL-1 and the GFP-MYC. We found there is a near 100% co-localization of these two types of transcripts, which means GFP-MYC reporter can faithfully reflect the endogenous distribution of IL-1. Therefore, we may use this line as a surrogate mark, we may using this line to map the brain-wide expression of IL-1. Okay, then we use MYC tag as a surrogate marker of IL-1 to look at which brain regions may express this receptor. We identified there are only a handful brain regions that may be positive of IL-1. One is the hippocampus DG area, and a network of Raphe nuclei, including the median Raphe nucleus, dollar Raphe nucleus, and Raphe magnus nucleus. To exclude a possible transient expression during development, we also verify these results using in situ hybridization in adult wild-type mice. So results are consistent with the previous MYC mapping. We can see hippocampus, dollar Raphe, median Raphe, and Raphe magnus are positive for IL-1 transcripts. Among all these brain regions, we noticed the DRN, the dollar Raphe nucleus, or DRN, is very interesting because DRN has been reported to regulate social behavior. And also its location is very special. It's localized next to the cerebral aqueduct, which may allow it to have direct access to all these soluble factors, including immune molecules coming from the cerebral spinal fluid. So we further characterize the IL-1 expression pattern in the DRN. The DRN is well known for its composition of different neuromodulators. So first, we want to identify do these IL-1 expressing DRN neurons express specific neurotransmitters or neuromodulators? We found over 80% of these IL-1 expressing DRN neurons also express 5-HT, which is a marker for serotonergic neurons. But very few, if any, of these I101 expressing DRN neurons express dopaminergic marker TH, which means these I101 expressing DRN neurons are predominantly serotonergic. And also, we use in situ hybridization in adult wild mice to confirm these results. We found, consistent with previous results, the majority of IL-101 expressing DRN neurons also express TPH2, which is serotonergic marker, but very few of them are co-labeled with TH or VGAT, which are dopaminergic or GABAergic neuron markers. So these results confirmed these I101 expressing DRN neurons are predominantly serotonergic neurons. Okay, next we just want to know, okay, these DRN neurons express I1 beta receptor, but do they indeed respond to I1 beta stimulation? We delivered I1 beta into the DRN, and then we examined the FOS expression in the DRN. FOS is a surrogate marker of neuron activity. We found that there is significant in the number of FOS positive neurons following I1 beta treatment, which means indeed I1 beta increased the neuron activity in the DRN. And also, we use a fiber photometry-based calcium imaging to examine the in vivo dynamic of these DRN neurons. And we can see, compared to vehicle infusion, I1 beta infusion induced a rapid increase in these calcium signals, which also indicate I1 beta administration increase the neuron activity of the DRN neurons. Now we have acquired the information like these. First, I1 beta, we identified there is a cytokine I1 beta, which can uniquely induce social withdrawal. And the neurons expressing its receptor I101 are identified in the DRN. And these neurons are predominantly serotonergic neurons. And using FOS examination and fiber photometry calcium imaging, we verified, yes, I1 beta can indeed functionally activate these I101 positive DRN neurons. So next, we want to conduct some functional manipulations. If our hypothesis holds, these I101 expressing DRN neurons indeed sends I1 beta signals and convert these signals into downstream behavioral changes, then experimentally activating these neurons should also induce social withdrawal, even without the presence of I1 beta. To confirm this hypothesis, we first need to label these I101 expressing DRN neurons with HM3D-GQ. So this protein is a chemogenetic tool, which can increase neuron activity following the administration of its ligand CNO or DCZ. We crossed these three lines, I101 Cre line and the serotonergic specific search flip line, and a third line expressing HM3D-GQ in the presence of both Cre and flip. We confirmed, okay, we are able to selectively label these I101 expressing DRN neurons with HM3D-GQ, and other brain regions cannot be labeled with this tool. And then we tested the behavioral changes following ligand DCZ administration. We found, indeed, increasing activity of these I101 expressing DRN neurons suppresses social interaction, and also activating these neurons can suppress the locomotor activity of animals. Next, similarly, we next, we need to ask, we have confirmed the sufficiency for these neurons to induce social withdrawal. But are these neurons necessary for IL-1 beta induced social withdrawal? To answer this question, we employed a similar strategy to label these IL-1 positive GRN neurons with HM4DGI actuator. So this actuator is also another chemogenetic tool, but its function is to suppress neuron activity upon the administering of its ligand. And similarly, we verified the specific labeling of these IL-1 expressing GRN neurons with HM4DGI. We found in wild-type mice, following IL-1 beta administration, they showed a robust social withdrawal. But with the mice whose IL-1 expressing GRN neurons are inhibited, they do not show robust suppression in social interaction following IL-1 beta treatment anymore. However, suppressing these neurons cannot rescue the IL-1 beta induced locomotor hypoactivity. So these results are actually very informative and important, because it tells us, yes, the activity of these IL-1 expressing GRN neurons are necessary for the manifestation of social withdrawal following IL-1 beta administration. However, they are not necessary for the IL-1 beta induced hypoactivity, which means the IL-1 beta induced social withdrawal and hypoactivity can be mediated by distinct neural circuits. So far, we have only talked about neural activity. Next, we need to ask whether IL-1 beta to IL-1 signaling pathway itself contributes to the generation of social withdrawal following IL-1 beta administration. To achieve that, we first knock out IL-1 protein in these GRN neurons. We cross IL-1 flux line with a third-degree line, and we examine the efficient knockout of IL-1 protein in these GRN neurons. And using this line, first, we found, okay, this knockout animals did not show baseline abnormalities in either social interaction or locomotor activity. However, after IL-1 beta administration, these knockout mice do not show social withdrawal anymore. This is in drastic contrast with its litamate controls, which showed a significant suppression in their social interaction. However, the IL-1 beta induced hypoactivity persists in both knockout mice and wild-type mice. Again, the dissociation between the IL-1 beta induced social behavioral change and this locomotor activity, this may indicate our social withdrawal phenotype is not a epiphenomenon of sickness-induced hypoactivity. Instead, this kind of social withdrawal is active neural processes driven by these GRN neurons, which express IL-1 beta receptor IL-1. Okay, so far we have conducted all our behavioral tests in an arena, and people may argue this is not a natural situation reflecting the natural social interaction within the animal cohort. That's why we employed another behavioral paradigm. In this behavior test, we co-housed 5 animals in a cage, in their home cage. And this home cage is equipped with 2 shelters. And Among these 5 animals, we designate one as a subject animal to receive the treatment of vehicle or I1 beta. After the cytokine administration, we observe how the social interaction between this subject animal and its 4 cage mates evolve over an extended amount of time. We usually record the behavioral change for 6 hours or even beyond. For vehicle-treated subject animals, we did not observe significant behavioral changes of these animals. They essentially spent almost all their time huddling with the 4 cage mates inside the same shelter. So this kind of social integration or huddling lasted almost throughout the full trial. But for I1 beta treated animals, we observe something very interesting. We can see this I1 beta treated subject animal isolated itself and remained in an unoccupied shelter away from the cage mates. And this is a more extreme or more strange example, because this I1 beta treated animal would rather stay in these open spaces just to avoid other cage mates. And more importantly, this is not random or transient phenotype. These self-imposed social isolation can last for extended period of time, even up to hours. We calculated the minimum distance between the subject animal and the cage mates to depict this phenotype. We can see vehicle-treated subject animals. The minimum distance between these animals and cage mates are almost near 0. And these fluctuations indicate randomly they will leave the group to eat some food or stretch their legs, but they will rejoin the group very soon. So these fluctuations are random and transient. But for I1 beta treated animals, we observe a prolonged and pronounced self-imposed social isolation starting approximately 90 minutes after I1 beta administration, shown in this red curve here. And also, when we activate I1 expressing DRN neurons using this chemogenetic tool, we were able to recapitulate this kind of prolonged social withdrawal, which means these neurons drive this kind of self-imposed social withdrawal. And also, when I1 beta is sufficient to induce this prolonged social withdrawal, inhibiting I1 positive DRN neuron activity can rescue this behavior. Now we know I1 expressing DRN neurons are both sufficient and necessary to mediate this kind of prolonged self-imposed social withdrawal. And also, we want to test the effect of I1 beta signaling itself. This time, we did not use I1 beta ICV injection. Instead, we used a systemic injection of LPS, which is a mimetic of bacterial infection. We found in wild-type mice, similar to I1 beta, LPS injection can also induce this prolonged social withdrawal. But in knockout mice, this kind of phenotype almost was abolished. We even use a real pathogen, Salmonella, and to examine Salmonella's effect. We can see before Salmonella inoculation, the behavioral performance between wild-type and knockout mice are very similar. But after the inoculation of Salmonella, wild-type mice showed this gradually increasing social distance between the subject animals and cage mates. And in knockout mice, this kind of social withdrawal is significantly attenuated. Okay, collectively, all these results showed I1R1 expressing DRN neurons are both sufficient and necessary for driving the social withdrawal phenotype. And if we delete I1R1 in the DRN to disrupt I1 beta signal in there, we will blunt the social withdrawal triggered by systemic immune activation induced by LPS or salmonella infection. Now we have identified these neurons in the DRN can be very important for social withdrawal behavioral phenotype upon I1 beta application. We also need to ask, what is the downstream neural targets? So what is the neural circuits mediate this kind of behavior? That's why we map the projection targets from these I1R1 expressing DRN neurons. Given that these neurons are mainly serotonergic neurons, we first look at the projection pattern of serotonergic neurons from the DRN. So I want to share this very beautiful work I found last year. They used a tissue clearing method to mapping a lot of cellular populations across the body. And this is the results for serotonergic neuron projection from the DRN. We can see all these white signals indicate the projection targets of these serotonergic DRN neurons. You can feel how wide expressed this can be. And we similarly conducted this projection target mapping. So we expressed TD tomato, we labeled these I1R1 expressing DRN neurons with TD tomato and mapped its downstream targets. Among a lot of downstream targets, we found several targets have been reported to regulate social behavior. So these targets are lateral septum, PVT, nucleus accumbens, central amygdala, as well as insular cortex. Then we use optogenetic to activate these targets or SOMA in the DRN. First, we shed laser on the DRN I1R1 expressing neurons. We found, consistent with previous results, optostimulation of these neurons can suppress social interaction as well as locomotor activity. And when we activate DRN terminals at LSI, lateral septum intermediate part, we were able to suppress animal sociability, but it does not affect animals' locomotor activity. And we also tested all other circuits from the DRN to PVT, to nucleus accumbens, central amygdala, and insular cortex. But none of them showed the regulation effect of social interaction or locomotor activity. So LSI can be a very interesting target to convey these information to further regulate social behavior during sickness. Now I came to the summary. So essentially, we identify a group of the DRN neurons that express I1 beta receptor I1R1. And upon peripheral infection, I1 beta level in the body increases significantly, which may trigger these neurons to increase the neural activity of these DRN neurons. And as a result, activation of these neurons may transform the animal's mode from social engagement mode to social withdrawal mode. We also identified the DRN2 LSI circuits may play a very important role in mediating this kind of social withdrawal. But we still need to address a lot of remaining questions because DRN has widespread downstream targets across the brain. And also, sickness behaviors are not limited to social withdrawal. It may manifest it as other behavioral changes. So does the circuit from DRN to other brain regions regulate other behavioral changes during sickness? This is a question we need to address in our future research. And I find the DRN to insular cortex circuit is very interesting because I found this paper. This paper published a few years ago showed the insular cortex can serve as a recorder to restore immune experience by reactivating this neuronal ensemble in the insular cortex. The researchers were able to recapitulate the immune response like scenario in the periphery. So insular cortex may be related to immune memory. And also, we found activating R101 expressing DRN neurons. We recorded a time-locked increase in serotonin release at insular cortex. And compared to vehicle treatment, LPS treatment can constantly increase the serotonin levels in the insular cortex. We do not know what these results mean yet, but this can be a very interesting direction to pursue. So last, I need to pay my acknowledgement to Gloria Choi, who is my mentor, and Jung Han, who is my co-mentor, and my collaborators, Matthias and Mario, who contribute substantially to this project. Okay, that's all for my presentation today. Thank you so much.